Good afternoon, everybody. Thank you for joining us. We'd like to thank Prof. Carlos for his time this afternoon. Prof. Carlos is a professor at the Northwest University, and this afternoon he's going to be giving us a talk on microbiology of groundwater, the good, the bad, and the ugly. Thank you, Prof. Carlos. I think you can share your screen, and we're looking forward to your presentation.
Thank you very much, Renier. I cannot share my screen because you are still sharing. Okay. Is my screen, can you see my screen?
Yes, Prof. Everything is fine on this side.
Okay, thank you very much. I'd like to thank the organizers for inviting me to present this talk. I'll jump straight into the subject matter. On the microbiology of groundwater. Groundwater has been for a long time perceived as being on the sterile side, that your surface and the fact that water percolates through soil and various layers, that the percolation sort of frees the water from microbes because of the size of the microbes. So this afternoon I'll be exploring a bit of that and use some examples from international studies to look at some of the good and the bad, as well as some of the work that was conducted at the Northwest University. Okay, so statistics that you are familiar with, that you are dealing with daily, that fresh groundwater is about 30% of the water available globally, and our surface water makes up less than 0.4% of available water. Quite a bit of focus has been on the surface water, particularly when it comes to the microbiology thereof. So this afternoon I'll be exploring a bit of microbiology of groundwater. So tapping into the groundwater sources could either be by boreholes or by natural eyes. And these waters become available then for utilization for whatever purposes we want to employ them for. Scanning the international studies, just a quick search through Science Direct and not even the other databases, using the terms groundwater and microbiology, it quickly provided me about 12,000 hits. I scanned the first 100 and about 40 of them, so about 40% specifically dealt with a microbiology of groundwater. Some of the others, the other 60 were dealing with other aspects of groundwater. What is demonstrated in these studies is that the microbial communities in the groundwater is dynamic. So that is the first point of departure, that there are changes taking place constantly. Many of the studies that looked at the microbiological communities focused on the risk from fecal contamination. And just now I'll be saying a few things about fecal contamination and how one can measure that. So there's a list of some of these studies, and if you just look at some of the topics, you'll see that it deals with some of the aspects that I've mentioned and highlighted in the previous slide. So I've taken limited number of these to show some of the work that is currently being conducted. And this is a study from an abandoned mine in Morocco, where the researchers have demonstrated that putting reed beds in the way of return flows to groundwater reduced harmful bacteria as well as other chemicals that enters the groundwater. And I've put a question mark there. Another study looked at the microbial communities in groundwater in response to an earthquake in southern Japan. And this study demonstrated that the influx of bacteria after the earthquake affected the microbial community structure. But a positive aspect that came out of it is that the denitrification potential of that aquifer was enhanced. A key to the denitrification is obviously the removal of nitrates. And is nitrates a problem in South Africa? In our groundwater systems, particularly in the northern provinces, and I'll show some examples of that. So once again, the question mark, potential technological applications to the situation in South Africa. This particular study focused on using water chemistry, isotopes, and microbiology to evaluate the groundwater sources and flow path. And they showed that this systematic approach of including the microbiology to infer subsurface flow was very useful at a regional scale. And once again, one should put another question mark there of a potential applicability. This particular study looked at sort of the combination of pathogens, and these were specific fecal pathogens. So they've used a culture-independent method where they've isolated DNA, and they've used the DNA only through PCR processes, where they've looked at the presence of various viral and bacterial and protozoan pathogens. The study clearly demonstrated that in these water sources in the US, there was sporadic fecal contamination of that sources. And the study provided a comprehensive data set, but interpretation and application of the data seems to be a challenge. And that is because of the analytical capacity that we have developed within the field of microbiology has outpaced regulatory framework. So once again, another question mark to that. Then obviously at this stage, with COVID, this was one of the studies, so the only study that I could find that sort of focused on COVID-19 lockdown, the impacts of heavy metals and microbes in shallow groundwater. The results showed that the shutdown of these industries, large as well as in small-scale industries, improved groundwater quality. This obviously had major impacts on the economy of the region, but in terms of the reduction of the hazard index, the hazard potential of that water was a clear positive. So they concluded that even though the economy was badly affected, that the results were useful when devising remediation strategies where prevention of contamination for an extended period could be a very useful strategy in ensuring remediation of groundwater. Then there was an example, a recently published paper by researchers from UNISA on groundwater contamination. The area that they've studied is sort of an overlap between Northwest Province, Limpopo, and Gauteng. And this study showed that from a physical, chemical, as well as a biological perspective, when these were classified, that many of these, or most of them, were unsuitable for direct drinking purposes. So this then brings me to some of the examples that we were involved in, research project from the Water Research Commission. This particular study focused on surface as well as groundwater within the Northwest Province. And I will only deal with the groundwater results. I'm not going to go into methodologies, and if there are any questions, I'll be happy to answer those. So threats on our groundwater sources is obviously population. Population density in red in the areas indicated in the red on that map of South Africa shows highly dense areas, and the highly dense areas could be anything from highly urbanized areas with lots of infrastructure to informal areas, sometimes occurring next to each other. And with an increasing population density, there is consumption, and with consumption comes waste. And waste has to be managed in some way, and much of it lands up in landfills. Sometimes, in most cases, what lands in landfills doesn't even have to be there. It could be recycled or be utilized in a different manner. People need to eat, and we are fortunate to have a diverse agricultural sector, particularly within our northern and central provinces, as well as in the southern province. Our northern provinces are also major production areas for our staple maize, as well as other crop types, and proteins such as cattle. So when you have these monocultures, obviously some sort of prevention of infection is needed. So pesticides are used, and antimicrobial substances to prevent these infections. But because once an infection occurs within an area, it can within a sector, it could lead to massive economic losses. And in some cases, some of these antimicrobials are used prophylactically to ensure that this doesn't occur. Then we are blessed with a diverse mining sector, and with the mining production comes lots of water is utilized within the production, within the mining processes, but it leads to waste. So all of these waste streams has the potential of seeping into groundwater from the agricultural sector, with lots of cattle and other species that is concentrated within a specific area. It means that from that could leach whatever treatment options are used, as well as the bacteria, viruses, fungi, etc. Could land up in our water sources. Minerals and metals, some of these metals are antimicrobial as well. I'm using the term antimicrobial. And that could also leach into the water sources. So these are some of the threats on the groundwater sources in South Africa. So the first example is drinking water from boreholes that are used untreated for drinking purposes. And we've used the SUNS 241 as a measure for the quality of the water, so we can classify the water. And that is in line with the World Health Organization's guidelines for drinking water quality. One of the important features of the guidelines, the South African National Standard and the guidelines is zero E. coli. Now, E. coli, Escherichia coli, is a bacterium that occurs in the digestive system of animals, and it is used as an indicator organism. So the question is always an indicator of what? It's an indicator of fecal contamination. If it is found to be present in water that is to be consumed by humans, then that water is deemed unsafe, because E. coli on its own, the species on its own, might not be pathogenic, but there are certain strains of it which are pathogenic. And an indication of fecal contamination could mean that there could be other pathogens that are present, viruses as well as other pathogens present in that water. So that is used as sort of a first step in testing for water. So the particular study in the Northwest Province, if you look at the map there, you will see that it's divided up into various areas, and we've grouped those areas based on sort of localities. So you have the most southern part here, J, the area J, which is close to the Northern Cape border, the Farlarts area. You have I, which is close to the Botswana border to the west, etc. So the values was for one season, and the numbers of samples that were taken is given there next to the codes that we used. If you look at the pH for SUNS 241 indicates between 5 and 9.7, and the water quality, and so far as pH is concerned, absolutely wonderful. The electrical conducticity was also way below the threshold limit of 170 millisiemens per meter. In some cases, the southern and the western part, the electrical conductivity was higher than the rest of the province, and that could be because of the geology of the area. Nitrates, in some cases, were very high. If you look at I and J, you'll see that it is extremely high, and in some other cases also very high. It should be below 11 milligrams per liter. That could have severe impacts on particularly infants and children under the age of 5. Then the biology part of it, the microbiology, the fecal coliforms and enterococci. In this case, we sort of looked at fecal coliforms as a group and not specifically at E. coli. We've later went on to demonstrate that many of that fecal coliforms are actually E. coli. Fecal coliforms and enterococci are from fecal matter, and we looked at the ratio of the fecal coliforms to the enterococci as well, and that ratio indicated that the potential of what we are seeing, the E. coli, were potentially from the from fecal contamination. The total coliforms is a sub group of the heterotrophic plate count bacteria. And heterotrophic plate count bacteria is this normal bacteria that occur in water, and you'll find it, you'll always find them, it will be there. And there are expert opinions that indicate that these bacteria may not be harmful, that they are regarded as harmless bacteria, and in many cases total coliforms are sort of used as indicators for processes when water is being produced. Total coliforms should be below 10 colony-forming units per 100 ml, and you can see it sort of exceeded that number on most of the occasions. Now, the question that we've asked is, are these harmful? And one of the studies that we've conducted was to take heterotrophic plate count bacteria from aquifers, and that was just from the Mooi River. We isolated the bacteria, and we identified them using standard microbiological methods, and we determined antibiotic resistant patterns for them. And bacteria that showed resistance to 2 or more antibiotics were then used, and these were fed to amoeba. So one species at a time to an amoeba mixture. What is anticipated is that the amoeba would then, through phagocytosis, ingest the bacteria and utilize the bacteria as a food source. What we did see, however, is that many of the antibiotic-resistant bacteria survived within the vacuoles of the amoeba, and we could re-isolate them and identify and show them that they were still exactly the same bacteria with the same characteristics that were fed to the amoeba. So what does that mean for us? Is that our white blood cells work exactly as the amoeba works. It's a phagocytic process where the bacteria should, once it lands inside the white blood cell, then it should be completely dissolved and broken down through the processes that occur. However, if these bacteria are resistant to amoeba, it means that they would potentially be resistant to the white blood cell processes as well. In another study, we've demonstrated that these bacteria, antibiotic-resistant heterotrophic plate count bacteria, had pathogenicity features, and I'll explain a little bit more about that in due course of what that basically meant. But for us, that was an eye-opener, demonstrating that the bacteria that are supposed to be harmless and that has been grouped as harmless might not always be as harmless as we think they are. We've also used cutting-edge sequencing technology to look at the microbiome of some of the boreholes. In this case, it was the Schweizer-Reinecke boreholes, the group of boreholes. From there, we've grouped the DNA, and we grouped the samples. We've isolated the DNA, and we've put them through sequencing processes. And the microbiome indicated that, yes, there were a lot of the bacteria that were present in the samples, whether it was in winter or in summer, that they are uncultured, and we don't know who they are, and we don't understand what they are doing there. But a large proportion in the winter samples, most of it, 70%, were uncultured bacteria, unknown, which could be useful. We just have to find out what they are doing and how they go about it, doing what they're doing. But the interesting aspect of this is that in this groundwater source, where we found these bacterial species, that we also managed to find sequences for E. coli, Enterobacter, Klebsiella, and Acinetobacter. Normally, when we do a microbiome analysis of a water source and surface water sources, the bacterial community is so diverse, and there are a large number of sequences from the other species that are present that we don't even pick up these indicator organisms. An important aspect of the species that we've picked up is that E. coli, or some of the E. coli species I've mentioned earlier, could be pathogenic, but Enterobacter, Klebsiella, Acinetobacter is part of the escape pathogen group that the World Health Organization has classified and said the escape bacterial group should be carefully looked at, because these are serious pathogens. We have to find and make sure that the antibiotics that is still available is reserved to protect us against these pathogens, because they could create huge implications. Klebsiella species, for example, has affected hospital, and it's particularly neonatal units that's been closed down in some cases. I know the KwaZulu-Natal had some outbreaks of Klebsiella, pneumonia infections amongst these neonatals. So it is a serious pathogen that could have huge implications. But amongst the bacteria that we've detected are a number of them that could be quite useful, could have technological implications that we in some cases know of, and in some cases need to investigate. So microbiome overlapped with the work that we've done with the culture-based methods. So this also overlaps then with the study from the scientists from the University of South Africa that demonstrated that biologically many of our water sources that are used for direct drinking purposes is actually not suitable for that. Then in another study, we have looked at drinking water, and that was, and I'll give one example of where drinking water, groundwater, is used for drinking water production. Two of the plants that we've used, in one case, the groundwater is mixed with surface water, and then drinking water is produced from it. In another case, groundwater, surface water, is mixed, which is then separately treated and purified, and then reclaimed water is blended into for drinking water production in that particular town. During the time when we were doing the study, in that particular town, there was no surface water, so it was only groundwater, and the reclaimed water that was available for usage. So the example, impacts, obviously, as I've mentioned earlier, could be those. So small town in the Northwest Province, water comes from an eye, and it runs through a canal towards the drinking water production facility, where a valve is opened and the water is taken in, and then it's pumped through to the drinking water production area, that house that is indicated there, to that house. And then in there is a rapid sand filtration, and then the water is disinfected and off it goes to the consumers. So we have sampled before the water goes into the rapid sand filtration, immediately after disinfection, as well as within the distribution system. Right, okay. So the land covers, what affects the eye is mainly agricultural land. So there's nothing really coming in that could have serious impacts on that water. So when we looked at the physical quality of the water, the pH was okay, TDS was all right, but insofar as turbidity was concerned, there was a bit of an issue. The raw water had turbidity less than the 1 NTU in most of the cases, but immediately after treatment, as well as in the distribution system, the turbidity was much higher, and we could then deduce that it was because of the rapid filtration process, the design of the system, and that was reported to the municipality for corrective action. Insofar as the chemical quality was concerned, nitrates was the main concern. There were other things as well, but that was sort of what sort of stood out. And then we looked at harmless bacteria, heterotrophic plate count bacteria. We've isolated them and we've identified them, and I'm not going to go into the species. But the numbers of individuals that were analyzed for antibiotic resistance to specific antibiotics, and there's a group of spectrum of antibiotics that we tested them against. There were 47 from raw water, and 23 from immediately after treatment, and 42 from the distribution system. And you'll see that the beta-lactams, ampicillin, cefalotin, as well as penicillin G was a major issue, as well as trimethoprim. And later on, I'll sort of refer back to those as well. So what we did was then from the heterotrophic plate count bacteria, is to select the bacillus species. Now, why we selected the bacillus species is that we found them present in the raw water after treatment, as well as within the distribution system. Now, bacillus forms spores, and spores are for survival purposes. So there were other work that we've also done, and which is not part of this presentation, which also showed that Bacillus species DNA was present in raw water after treatment, as well as within the distribution system. So we've selected isolates, we've isolated the DNA, and we've sequenced the entire genome. And from the genome, we observed that what appeared to be sort of a selection of these genomes after treatment, that the genomes and diversity of those genes were higher after treatment than within the raw water. But I think important here is that multi-drug resistance genes were the ones that sort of dominated. Right on top, you see the trimethoprim genes were also present, and a number of other genes that are that we have seen resistance to in our when we did the actual studies for the antibiotic resistant patterns. So that sort of overlaps with it. Then the pathogenicity features, which I previously referred to. These are extracellular enzymes that are produced by pathogens. Proteinases breaks down the proteins, lactases the same, lipases the fats, and DNases DNA. And that is to either cause severe breakdown of these cells. The hemolysis test is when you grow the bacterium on blood agar, and you observe clearing zones, which shows that the bacteria is actually breaking down those the red blood cells. So a large proportion of the bacteria that we've used had or produced more than one of the pathogenicity features, indicating that these bacteria, generally regarded as harmless, that will be occurring within these water sources, might not be as harmless. We've seen similar patterns within those that survived that we find in the drinking water. The features that we have seen sort of overlaps with the whole genome, because from that same whole genome study, the same isolates, we could see the virulence genes that overlaps with the features that we have seen. So the genes were present, they were present within the bacteria from raw water, the water after treatment, as well as within the distribution system. Another thing that we did was to measure antibiotic residues within the groundwater using fancy chemistry, and indicated there the ciprofloxacin as well as the penicillins were present, and there's a neomycin that was also present that were detected. The trimethoprim and colistin, as well as beta-lactams, we did a specialized test where we could quantify them. They were present, in all cases, they were present in these water sources. Now you see there's a predicted no effect concentration, which is in red, and it's very high, indicating that the presence on its own in the water will not allow for selection of antibiotic resistance. But the question is the combinatorial effect of all these antibiotics that could be present, as well as other chemicals and metals that are also antimicrobial substances. What are the effects of that? And that's something that we are still grappling with, and there's a lot of work that is going around internationally looking at that. But what I'm saying is that from this particular study, and there's some of the others as well, where we have seen low concentrations of these antibiotics present in that groundwater, which indicates that there's some sort of usage of these antibiotics. And colistin is one of the antibiotics that is reserved for treatment of serious human infections. Beta-lactams are constantly used, and many of us are using it, the penicillin, ampicillins, etc. But it's also used in animal production. So trimethoprim is an antibiotic that is used in conjunction with kanamycin and some other antibiotics for the treatment of infections in the urinary system. The results of this is within WRC reports. So that is available for scrutiny, and it has the particular systems that use groundwater for drinking water production, and the system that I explained, the example that I've used, might be used in many other parts in central South Africa, central and northern South Africa. So similar types of results might there be. Some of this was published recently, so that is also available. Now, this particular 2019 article in the Daily Maverick sort of gives an indication of why it is important that this focus on our groundwater is placed, and that quantity linked to quality, but not only physical and chemical quality, but also the microbiology of groundwater is important. And just this morning, there was interviews with the Gqeberha municipality representatives about their strategy to use groundwater to supplement the water from the city or from the metro. So very important that this becomes part of our agenda. So from my side, the take-home message is that this groundwater is a valuable resource, but it's not free from microbes. The bacteria in the groundwater may be from fecal origin, and others that are regarded as harmless may not be as harmless as proclaimed. So treatment is definitely essential. The filtration of antibiotics as well as other chemicals, metals, etc., into groundwater is of concern because it affects the usability of that water. The antimicrobials that could lead into the groundwater could select for resistant bacteria that occur within that water, affecting the usability again. The microbiology of groundwater is dependent on surface processes and links to these surface processes, as well as the geology, the geochemistry, seismic events, etc. Anthropogenic activity may have negative impacts if protective measures are not put in place. There are some examples that I've mentioned earlier on, which could potentially provide some sort of technology, low-cost technology that could protect our groundwater sources. The microbes in the groundwater is potential technological applications, but we need more studies to investigate that and to determine what sort of applications is possible. The protection of this important resource, 30% of the available water globally, is critical from a chemical as well as a microbiological perspective. So some of the individuals that were involved in this studies, given there, and then I want to thank everyone for listening to this, as well as funders, Water Research Commission, as well as the NRF for providing funds that made this possible. So thank you very much.